Hi all, welcome to session 10 of my ozone technology course. The session 10 will introduce scale formation, RNA, DNA, the structure of the virus, its function, its destruction, and some information on cysts. Scales are deposits, generally white, sometimes creamy, and if the water contains iron, it will be about light brown or dirty brown. The scale usually contains calcium carbonate. When water concentrates, the totally dissolved solids increases. It will react with the sodium hydroxide that is resulting from the disinfected added to the community water and therefore form insoluble carbonates. Eventually, it will become saturated and it will deposit or crystallize as scales. However, the scales can form subject to certain conditions. The organic matter in the water, which will stick onto the surface of the water, forming slime. It also initiates the crystallizations of the calcium carbonate to form scales. The organic matter is resulted from the dust and all other particles that will settle onto the water from the air. The bacteria now use the slime as food and accumulate within the slime. If slime is absent, calcium deposit will spontaneously crystallize and settle as white deposits of scale. But if the slime is present, then the scale formation is encouraged. The deposits are layered one on top of the other. They are porous and bacteria can move from one part of the scale to the other part, and they are able to survive even reasonable disinfectants that may be added to the water. The issue of scaling will be discussed more in the cooling tower sessions. This slide shows photographs of scales. If you look on the left picture, you will find that the scales had almost blocked most of the chiller holes. This is actually the phase pipe of the chiller pipe that has been opened. It's a chiller plate of a heat exchanger. When that much of scale blocks the holes, then there is insufficient flow of water going through, either through the chiller, particularly through the chillers, and therefore there is a reduced heat exchange, and the air conditioning system, if it is being used, will break down. Now if you look on the right top picture, you can see a hot water pump or hot water pipe. The amount of scales that can accumulate and virtually block almost the whole pipe. If you also look at it, you will find they are in layers of different colors. This is actually the deposits over a period of time, and each time period is a layer. If you look at the picture behind, on the bottom of the right, in the left you will find the scale deposits on the plate on the sides of the pipe, and if you look at the pipe on the right, it is after cleaning the pipe. So obviously you can remove the clay, all the scaling through two processes, either physical scrubbing and scraping, which of course costs time and money for labor, and secondly, you could probably use acids to wash and slowly dissolve and react with the scales. However, caution must be exercised when you use acids, because the same acids that will remove the scales can also attack the walls of the pipe and eventually corrode to a level it reduces the thickness of the pipe. We shall now discuss RNAs and DNAs. RNA, the definition of RNA is ribonucleic acid. The definition of DNA is deoxyribonucleic acid. Ribonucleic acid is a strand or a chain of compounds that are formed by the combination of several different types of nucleic acids. Now, there are a few types of RNAs, and each RNA has got specific functions. These RNAs and DNAs are found in the cell of an animal or a plant. You find that the DNA is actually a combination of two of the RNAs, where one sugar part is used for the joining, and that is why you call it deoxyribonucleic acid. The RNA will serve as templates for the manufacture of DNA chain. The RNA is also important in various other functions, as we will see in the next pictures. Just for example, COVID virus includes SARS, MERS, and COVID-19 appear to be RNAs with an envelope of a protein sheath with protrusions that resembling crowns, hence corona. These virus and the RNAs that are in the virus duplicates itself within the cell of the animal or a plant. If you go into the picture on the right, you will find that a ribosome, which is also a type of combination of RNAs, is capable of attaching two RNA chains to form a DNA chain. In the right, you can see the tRNA or transfer RNA, which actually connects or collects a specific nucleic acid and brings in and attaches to the template of another RNA template and makes a connection so that the amino acids or nucleic acids that is connected to the DNA, tRNA can be combined to form another RNA chain. This slide shows the structure of the virus, and the picture will also show the structure of a DNA and RNA. DNAs and RNAs are found in all animal and plant cells, and they are very vital in the metabolism and survival of the plant and animal cell. The virus is composed of a nucleic acid based chemicals forming the DNA RNA chains. This is the structure of the RNA of all living cells, and the RNAs and the DNAs are of different composition. They are formed from blocks, building blocks of 4 of the nucleic acids: cytosine, guanine, adenine, and thymine, usually called CGAT. So one block can consist of CGAT or CCGT or CCA, etc. So that will be a building block. Now, each of these building blocks are combined together to form an RNA chain. And even the building blocks are in different orders. And because of them being in different orders, differentiate one animal cell to another animal cell or a plant cell. It also differentiates from a human, monkey, fish, etc. The order of the nucleic acid bases are critical for the characterization of the DNA or RNA. If the order or the composition is changed, then the DNA will lose that particular character. Please have a close look at the picture on the right. It will show you both the RNA and DNA chain and how they are combined from the colors. We say within the chain you will find that there are colors that will show each of the nucleic acids. We know that there is a big virus infection going throughout the world, and this infection is by the coronavirus or COVID-19. Out of interest, I have taken a picture of the virus and the structure of this particular virus. If you notice that on the left you find the whole virus, which is a big round ball-like structure and protrusion of spikes, which are proteins. Now, if you look on the left, you will find the inside of the virus, which contains the RNA protein. The RNA protein is the one that is the active part of this virus, and when the virus sticks onto the soft tissues or the cell of an animal, in this case our human cell, it is able to combine and penetrate into the cell. The protein breaks down, that is the wall of the cell of the virus breaks down, and the RNA protein goes into the cell, and that will encourage the animal cell to reproduce and form more of the RNAs, as I explained to you in the previous slide. If you observe the spikes, you call it a glycoprotein. The wall of the virus, which is an envelope, it is also a protein, and the RNA, it is also a protein. Now, as we all know that virus on its own, that is the RNA on its own, it cannot multiply because it is a chemical. But the minute it goes into the cell or host cell, then it uses the host cell to give instructions to the host cell to manufacture copies of itself. And that is how the virus multiplies. And obviously, the virus itself can cause collateral damage onto the human systems. You will also note, when we come to application of ozone, ozone will react with any double bonds or triple bonds. It will react with any sulfide groups, any nitrose groups, any acetyl groups, and is capable of breaking them down instantaneously. And once any single link is broken, then the virus will no longer exhibit its characteristic, and in fact it may be totally infertile, or this slide shows the structure of the MERS virus, and on the right the SARS virus. If you notice, they look very similar to the present COVID-B. This slide shows in writing what I have explained in the previous slide. This is how the coronavirus infects the body. The virus enters the body through the inhalation of air and through physical contact, through atomized particles or body fluids. It gets absorbed into the body and penetrates the cells and use the facilities of the cell to multiply its RNA. The multiplication is very similar to the multiplication of nucleotides, cells, etc. of the human cell or animal cell. The virus generally does not remain viable in the air for long unless it has a protective shell, which lengthens its activity time. The coronavirus has a protein sheath which protects the RNA of the virus, and it also facilitates the penetration of the virus into the cell. The protein is shed, the RNA enters the animal cell and multiplies. It creates a sheath to be exhaled in the body, or in the body fluids. The coronavirus remains viable if the humidity exists and for some time. The protein sheath should not dry up or get denatured. Please note, ozonation can cause denaturation of the protein sheath, breakdown of the spike proteins, and render the coronavirus inactive. This slide provides some information of what cysts are. Cysts is actually a result of a cell or bacteria, for example, which develops a protein sheath, very similar to the virus RNA having a sheath on itself. Those bacteria are generally known as tropozoites because of their different shapes and looks. Now, there are two aspects of the cyst. There are many types of cysts. I have taken only a Giardia cyst, which is very common in our drinking water. You might recall that Sydney water had a big infection of Giardia, which caused quite a number of gastritis to people who were drinking the water, and they had very much difficulty in trying to get rid of the cysts. Tropozoite forms cysts and becomes stable, and once it enters the human gut, it sheds its shell and becomes an active bug or bacteria that will cause your damage. Here again, normal bleach or chlorine that added to water will not be able to destroy the cyst. Ozone can cause some damage, but it's still not strong enough to be able to destroy or get rid of the Giardia. But ozone, in combination with other oxidants, can effectively remove Giardia from water. This slide contains an abstract by Rowan MD and Robbins DPM, and the source is from Journal of Infectious Diseases and Epidemiology. it is very clear that application of ozone can resolve much of the infection situations, but it is not being used at all. It's obviously, as I did explain, that ozone therapy is not expensive at all. It is safe, and it can destroy all the virus, inclusive SARS and COVID too. The question is why the drug companies or other researchers do not want to apply ozone technology is obvious. There's no money to be made. For those who are interested in further information, please look into structure, types and functions of RNA and DNA, overview, uses and side effects, precautions, etc. And you can also see quite a number of YouTube information on the effects of COVID on human beings and other viruses. And if you look into effect of ozone on viruses, you will find quite a number of articles which will establish that ozone is extremely effective to control the virus infection. Yet nobody is using it. And the answer is obvious. We have come to the end of this session, session 10. Thank you for watching my video. Please proceed to session 11A, which will deal with the manufacture of ozone by UV, application of UV light for disinfection. If you are new to this channel, please refer to sessions 1 and 2 that will explain the course program, and 3 to 9, which will be the program. If you like the course, please tick the like icon and the subscribe icon when you will be notified as soon as further sessions are released. I thank you and have a good day.